Hi! It's time for us to have a fine discussion of the topic of genetic variants. How do we go about finding them, and how do we go about figuring out what they do? Today's lecture is going to cover a little bit about the nature of genetic variants, specifically which technologies we use to go hunting for them. We'll be comparing whole genome shotgun, whole exome sequencing, and microarrays for that purpose. We'll then talk about how we evaluate the impact of a particular non-synonymous SNV. We'll get into the definition of that term as we move along. In terms of phenotypic impact, when we have a change within a protein coding gene, how do we evaluate whether it's deleterious to the activity of the gene product? And finally, we'll move into genome-wide association studies, which were at one time hugely in vogue, perhaps slightly less so now, for determining which genetic markers that we have in terms of SNPs are most predictive of someone's taking on a particular phenotype, for example, a disease risk. So we're going to start with whole genome shotgun and mapping. Now, hopefully alongside this, you've also seen the video on genome sequencing, mapping, and assembly. So the concept of mapping should be familiar from that. In the case of variant hunting, we are almost always looking for sequence variants that are different from, that stand apart from, what the reference sequence says should be present at that site. So this is a case where we have lots and lots of reads from our sequencer, and we now need to map them to the annotation. But we need to allow for a mismatch of a nucleotide or two, for example, to allow us to find variants, not just the bits of sequence that align perfectly with the reference. Okay, so you might expect that individual humans are going to vary from each other to some extent. I believe that the most recent estimates I've seen suggest that a given human a pair of humans drawn from the world population will be genetically identical to the level of 99.5%. But that's still a fair bit of genetic marker to differentiate one person from another. And as you might imagine, differentiating one person from the reference genome is also going to have that kind of metric to it. So you might expect then that an individual person for whom you've run whole genome shotgun will differ at more than 2 million sites from the reference genome. This is largely because we have just a tremendously large genome, something like 3 billion base pairs. So there's a lot of room, even if you have a variant every 1,000 nucleotides or so, that you're going to have a very large number versus the reference sequence. Okay, so it may be that you're looking for a variant that's associated with phenotype, and you don't see as much sequence coverage as you need to in a given gene in order to find that variant. What if you had only, say, 5 reads that mapped to a particular gene, and this person was heterozygous at that site? Well, it might be that 3 or even 4 of the reads were for the wild type sequence and only 1 or 2 were for the variant sequence. That's really going to be a very small amount of evidence on which to claim that this person contains this particular variant. So necessarily, if we're doing whole genome shotgun sequencing as a way to evaluate which markers a person contains, we're going to want to do very deep sequencing in order to get a very deep coverage as far as we can on all genes. Okay, it is possible, however, that you may do the same sequencing experiment on the same samples multiple times and get different results. A lot of people today treat sequencing datasets as though they are full of all information one might hope to get and that they're perfectly reproducible. This is not the case. Sequencing experiments still give us somewhat different readouts when we repeat them on the same samples over and over again. This is particularly the case when we're looking at insertion and deletion markers. These are to say places where we've added or subtracted some of the nucleotides from the sequence. So it may seem like whole genome shotgun is a dropkick. Of course, every patient should be sequenced, etc. Even if you set cost aside, there are technical variances at play that may limit your ability to detect the same changes every time for the same sample. one of the ways that we frequently go in pursuit of rare sequence variants, of mutations believed to be causative to a particular disease, is the use of triads, which is to say that we have a father and a mother and we have a child who is affected with some disease. In this case, we're looking at something that we would describe as autosomal recessive, that the father is a carrier for this allele and the mother is a carrier for this allele, but both of them are asymptomatic. The child, however, who has both of his or her, in this case, his alleles that are the disease, the disease-causing one, has the disease, and we can use the parents in this case as a point of reference for evaluating which of the genetic variants present in this affected child may be disease-causing in this way. So there are lots of ways that people have talked about using triad sequencing for this type of rare disease thing. So we start with the fact that a lot of people have recognized for a long time that comparing a child to reference is not going to give you a lot of information because you're going to have essentially too much data to have information. That you're going to have, as I've said, 2 million variants to choose from, and trying to decide which one of these is pathogenic is really problematic. The parents are in this case very useful controls for that, to be able to say that the genetic variant that this child has is special. In this case, it is homozygous, where the parents are heterozygous, for example. So using the parents as controls is very valuable for this. Comparing against a world-derived reference annotation for the human genome is going to be a lot more remote than the parents will be in relatedness. So we're able to throw aside huge numbers of variants present in this child because they were also present in the parents and weren't disease-causing in them. Okay. It also may be that we see preferential transmission of alleles, that if you see a large number of triads associated with some particular disease, we see that in particular cases we're seeing a preference that one allele is transmitted to children over the other. It could be in this case that it has a survival benefit, right? That if you have an allele which, if present, reduces their chance of growing to maturity, that's going to be strongly selected against. So in cases like this, we see that this triad sequencing gives us the ability to detect whether these variants are continuing on to further generations or not, and narrowing down our focus on which ones are of interest. I would also point out a class of algorithm called a phasing algorithm. There are software out there like the ShapeIt algorithm that allow us to determine haplotypes, which is to say, given that a given person has this set of variants, which of them have come from which parent? So if we think of a given variant as standing alone, we lose a lot of power because it may be that this variant and another variant in the neighboring gene is always transmitted as a pair. In a case like that, understanding that they are related in this way, that they are a haplotype and not just independently observed alleles, is actually quite a lot of value. So this is the process of phasing. We're not going to discuss it in a lot of detail in this talk. In all of these cases, though, triad sequencing is valuable because in understanding an offspring's genotype is most capably done when we have the context of the parents to work from. So having talked about some of these broad challenges in spotting these variants, it stands to reason that we would want to discern which variants are chance observations, maybe the sequencer was just having a glitchy day, and which ones are very strongly evidenced. So algorithms like VarScan 2 have been created for that purpose. So what if we have sequencing errors? Right? It's plausible that if you found a subset of variants that you thought were really interesting, you could go about doing Sanger sequencing, targeting just the region where they appear in this person, to confirm them by a follow-up experiment. But is it possible to learn algorithmically which of these variants are real? So we start with sequence coverage. We've already talked about this as a problem in whole genome shotgun, that if you don't have enough sequence coverage in terms of how many reads pile up at a particular nucleotide, it may be very difficult for you to say with any clarity whether this variant is real or not. Ideally, you'd have 50x coverage everywhere, but that's simply not the case. So being able to evaluate where sequence coverage is sufficient for us to make a decision is important. Next, base calls. A couple lectures back, we talked about Phred scores, which is to say, what is the chance of error for each of the letters that we've called in the sequence? If you're making your claim that a particular variant is present in this person, and the evidence upon which you're basing that is very weak, that it was a really lousy signal on the sequencer, you're in a much weaker position. So being able to evaluate the base call error probabilities is quite important. As we've said, many reads must attest to this variant allele. If you're dealing with a heterozygous allele, one that is only half of the genetic material that this person has, attests to this less common sequence variant, then that's going to diminish the amount of evidence upon which you can make the call. Of course, if you've got a variant that's lethal in homozygous condition, that's certainly worth knowing as well. Okay, so you want to have as many reads as possible to determine whether you believe this variant. And of course, as is true in almost every bioinformatics algorithm out there, a statistical model needs to be in place. And so VarScan 2 provides this statistical model for evaluating whether the cases and the controls differ at a particular variant or not. Now, one of the outputs that we get from this is a really common type of file format that I'd like everybody to know about, and that is the VCF, or sometimes called a BCF, the binary version of this. This is a variant call format file, which is to say that instead of giving someone the raw output of the sequencer in, say, FASTQ format, instead of giving someone an annotation in FASTA format, or an alignment of the two in SAM or BAM format, we're giving someone a very short, a very abbreviated version of what we found. Basically, this is the roster of sites that were detected to be variants in this sample. So in this case, we have some header information up at the top. Here I'm using an example I've drawn from Sam Tools GitHub IO HTS specs. This is the website that describes the VCF format. There's also a really useful paper from 2011 that defines this format and its uses in a lot more detail. But here we see that we've got a header that specifies the file format itself, the date it was collected. We have the reference, which says to which sequences did we align reads in order to find these variants. And it has contig information that describes the data that have been assembled out of these reads. So we have several different fields that are present here. I've actually abbreviated one, the info field, because it made this utterly illegible. I realize on camera you're not going to be able to see this, but again, you should download the slides from the link that's provided below this video on YouTube. What we see is that we have information about the very broad scale location, here chromosome 20, the position within that chromosome, an ID, because very frequently the variants that we see are attributable to SNPs, very commonly known polymorphisms that are present in a large fraction of the community. So many of the sequence variants that we see are recognizable to existing SNPs. What is the reference letter that should be at that position? What is the alternative that's been seen by the sequencer? What is the quality score associated with it? And in this case, we have a filter that's been applied to say that that some of these variants have been quality checked and passed, and others have failed. So we can see that the first one is listed as a pass. The second one, however, failed because of the Phred score quality filter of 10. Its score was too low with a quality of 3. We see some format information, and in this case, we are specifying the variants seen across 3 different samples, not just one. So VCF is a pretty compact way to represent not the full assembly, not the full mapping, but rather which variants were observed in this particular sample versus that annotation. Okay, right. Now helpfully, people have been looking at variants quite a long time. Big efforts like the HapMap project did quite a lot to sequence people from many different ethnic groups to determine which variants were visible within those populations. A great many of these databases have now gone on to incorporate huge amounts of exome sequencing, a topic we're going to get to next. But I want to start with a common bit of language that we still see in a lot of literature, some of which I think is starting to feel a little dated and confusing. So when we talk about SNPs, we're talking about single nucleotide polymorphisms, which is to say that we're not making a broad statement about a huge piece of DNA. We're talking about an individual position, and generally speaking, a SNP is going to be one letter replaced by another letter. And that can be either a transition or a transversion, depending on the biochemistry there. But this single nucleotide polymorphism is a type of sequence variant that's actually quite common in the population. Back when I was in grad school, people would say that in order to be a SNP, you had to be present in at least 5% of a population. As I read a more recent article on this subject from 2015, people were saying, well, really, these days, we use 1% as a cutoff. If 1% of the population has this variant present, then it's a SNP, and if it's less than 1%, we would call that a mutation. Unfortunately, people have a lot of mistaken attributions that they put on these terms. A lot of times people would say, if it's just a SNP, it's benign. It has no effect whatsoever on expression, no effect whatsoever on the gene product. This is not, however, accurate. I would ask you to think about the example of sickle cell anemia. Sickle cell anemia is as I recall, a hemoglobin variant that is present in more than 1% of the global population. If people are homozygous for it, then yes, they have a really dangerous disease. But it has some protective advantage if it's found in a heterozygous condition. So in a case like that, we have a SNP because it's commonplace in the world's population, but it is a deleterious SNP, especially if found in homozygous condition. So it's not always the case that a SNP is benign and a mutation is automatically pathogenic. We're going to have a lot of discussion in this talk about how to evaluate whether a particular variant actually causes disease or not. Okay, so at the moment we're kind of setting this boundary at about 1%. If it's in more than 1% of the population, we call it a SNP. If it's very rare, vanishingly rare, we might call that a mutation instead. More generally today, we try to be a little less judgy about the language we use to describe these variants. We just call them genetic variants. This is the recommendation of the ACMG. So these older terms, SNP, or polymorphism, and mutation, are still going to crop up in quite a lot of the literature. These have now been stored in databases like dbSNP. The 1000 Genomes Project has developed good allele frequencies for the many genomes that have been sequenced there. The Exome Sequencing Project, the Exome Aggregation Consortium, and both of those sources have been jammed into the Genome Aggregation Database, or GNOMAD. So we have a variety of these resources we can call upon when we discover a SNP, or discover a variant, to determine whether it's been seen before, and if so, in what fraction of the population. ANNOVAR is a really valuable tool for us to start digging through the great heaps of sequence variants we detect anytime we sequence and map a particular person's DNA. So ANNOVAR is going to receive a list of the genetic variants, what positions they take in the genome, what is the observed letter, what was the reference letter supposed to be, and from that, it can do quite a lot. It can detect protein coding changes, which is to say, this sequence variant appears within a protein coding gene, and it causes a change in the amino acids. Very important. It can also recognize sequence variants that fall within a particular genomic region. It might be that you are really interested in discerning which part of this region of DNA is, essentially, laying onto your list of all variants a very narrow filter to say, I'm only interested in stuff that takes place in this particular genetic region. Maybe you have a region that's been duplicated a lot in that particular genome. Things like that. Okay, you might check for the presence in databases. People have made these databases for this purpose, so being able to say what's novel for this person, what hasn't been previously observed, is actually quite powerful. One of the most frequent uses that we see is kind of an alternative usage of the software. We might want to develop candidate gene lists for Mendelian disease. So if you have a disease that you believe is caused largely by the genetic variants that a person contains, you can use this ANNOVAR software to help you narrow down the list of which variants are most likely to be of benefit to you. Over at the right, we have this flow diagram showing, in this case, 4.2 million SNVs that have been detected, and 500,000 insertions and deletions, here narrowed all the way down to 20 genes that are believed to be, that may be associated with this particular phenotype. Looking through 20 gene the variants of 20 genes is a lot easier than looking through 4.2 million. Now, helpfully, Brigitte Glanzmann in our own department has created some software that leverages the output of ANNOVAR to help us get down to a gene priority list, to be able to prioritize which variants are likely to be most useful for this. Now, her paper was published back in 2016, so you can see that starting from ANNOVAR, she's able to do things like removing synonymous variants. You know, if you don't change the amino acid sequence, it's a lot harder to assess that this variant actually has any effect. It's able to do some additional filtering against the 1,000 genomes and so on to cut down this space. So quite a lot of power, and here at the bottom we have this interest in Fathom scores. Fathom is a tool that allows us to evaluate phenotypic impact. We'll discuss that in the second part of today's talk. So we talked about whole genome shotgun. What about the exome? Are we sequencing something substantially different if we work from the exome? In whole genome shotgun, we're grabbing bits of DNA from all over the genome. Exome sequencing, on the other hand, is enriching for particular targets. In this case, the protein coding genes most typically. So this is very helpful because the sequencer doesn't have to sequence everything. We're looking at maybe 1% of the total genome. That allows us to focus the attention of the instrument, giving us reads that are reflective of the expressed genome, not the genome at large. This gives us more read depth, which can really improve our sensitivity to recognizing variants that are present in particular places. This is, again, hugely valuable for that. And it becomes easier if we see a variant from exome sequencing. We have a much better shot of being attribute it to phenotype than if it's a variant we've seen in whole genome shotgun instead. But as you can see, these steps are going to involve some sort of hybridization with an enriching agent. In this case, we have capture probes in here that represent streptavidin biotin chemistry for pulling out all the sequences that are that match well to these probe sequences. These probe sequences, in turn, represent the protein coding genes. Now, there's still a fair bit of variability that we have to deal with in this in exome technologies. We would start with the fact that depending on which enrichment kit you're using to enrich for the exome, you may get you might get a slight difference in which genes are included, and it might be that different introns of those genes are captured as well as the exons. If studying markers within the introns is of value to you, exome sequencing might be a real push for you because it may be that the amount of information you get from introns is too limiting. It might be that you're interested in microRNA or promoter sequences or ultra-conserved elements. It's frequently the case that these capture kits will not include genes of that sort, features of that sort within the set. And probe efficiency can vary as well. It may be that you're grabbing out some sequences that don't in fact map to the annotation in the end. So it's really worth understanding which capture reagents are going to help you see the particular genetic regions you're interested in. I would also note that there are a fair number of people who've taken this argument even further. We started with whole genome shotgun where we were sequencing everything. We shifted down to just the protein coding genes, just 1% of that total space, with exome sequencing. And now we might say, well, I'm not actually interested in all 20,000 human genes. I might only be interested in about 1,000 of them, or just a few hundred of them. For cases like that, you might use something like AmpliSeq. So at our at the Stellenbosch University Central Analytical Facilities, we make use of a thermo-ion proton and an ion S5 sequencer pair. So these instruments are really great, and we can use them with these AmpliSeq kits from Illumina if we're trying to focus on a particular subset of the genes. So I've given the citation for a really recent paper here that came from our genetics folks. What they were able to do is focus on 751 genes that are known to be associated with neurological disorders because their study was in Parkinson's disease. So it's possible that there were other genes that were also of interest to Parkinson's, but the 751 in this community kit was already a really good sampling of very high probability contenders for genes associated with Parkinson's. So they were able to evaluate 47 people from here in Nigeria who had Parkinson's disease and recognize a pile of new sequence variants that they could associate with Parkinson's disease. I would remind you that microarrays are still around, and if you don't have to deal with a sequencer, if you can just run a microarray or a bead array experiment, you're going to cut your costs quite a lot. You're going to cut down the volume of data that you produce, which is going to make it not only easier for you to store it, but also to analyze it properly. So I would note that there are a world of these microarrays and bead arrays still being produced, and you can see that the cost can be as little as 1/10 of what you would pay if you were having to do sequencing of everybody. These bead arrays are going to give you a limited number of markers by comparison to an exome sequencing, or certainly than a whole genome shotgun sequencing. But they're still quite valuable and give us a lot of information for a lot less money. Now I'm going to take a brief moment to step aside from the technology of it all and talk a little bit about our responsibilities as scientists. That might seem pretty weird from an information scientist kind of guy, but the reality is the information we produce through sequencing experiments is very important. And the way that we handle it can potentially expose us to lawsuits. So it would be a good idea for us to take just a moment to think about privacy and identifiability in our data. So why do we need to take care about publishing DNA variant data? This little diagram that I found in ACM computational surveys I thought was really valuable. We start with health and behavior. DNA is predictive of someone's health, right? We can predict with pretty good accuracy who's going to develop certain diseases and who isn't, right? So that is certainly a reason to protect the data. I've written here that a person's genome does not change much. Here we're using the principle of that DNA is static for a person, that it's traceable for a person. If you find a bit of someone's DNA at a crime scene, for example, that has very severe personal consequences for that person. So generally speaking, throughout the course of a person's life, their DNA doesn't change a lot. If you're looking at tumor DNA, you're obviously going to find lots of changes associated with cancer. All right. A genome is usually unique for a person. If you're an identical twin, perhaps that's not the case. But generally speaking, DNA is able to pinpoint you out of the world's population to a very great extent. We can move on to the concept of mystique. At present, at least, in the media, in public perception, DNA evidence has a credibility that is often lacking in other kinds of evidence that we see at trial. And generally speaking, this special standing means that there's a spotlight of attention on DNA variants. We should not assume that no one cares whether DNA information is provided or not. Sequencing data are perceived to have special value. We spend a lot of money to get it, And retaining those data is also treated as a very valuable proposition. Giving that information away for an unwilling person is to give away something of theirs. So this value is a big concept. And biological kinship. You know, we all may feel that we have great certainty who our grandparents are and maybe even who our great-grandparents are, but the reality is, if we were to look at the DNA of people and their presumed family relationships, we could upset a lot of people. So we don't just release DNA for people willy-nilly because it can have very serious consequences for their family relations. Okay, so you think to yourself, if that's the case, all I need to do is just cut off the names of my database and then I can dump it into the public. People will often say that this is de-identifying the data. There are a lot of weaknesses to this. I'm not going to go through all of them, but I would recommend that you take a look at these two papers down at the bottom. This paper by Sweeney et al. Did a lot to motivate the NIH to create its safe harbor specifications for basically how do you avoid getting yourself in legal trouble for publishing your data, right? So it's not just enough to strip out the names. We start with this concept of identifiability. This is the degree to which materials stored in biobanks, including data, can be linked to the name of the individuals from which they were derived. That's a broad definition here of identifiability. I thought this quote was actually really informative. Only about 100 SNPs are required to distinguish an individual's DNA record. You might think, okay, well, if I have just this one variant that I'm publishing, that's not 100, I can publish it. But what if only 0.0001% of Earth's population has that variant? Suddenly you're looking at a very small number of people who could possibly be represented by your study. But even if you're just talking about broadly popular SNPs, right? What if you're looking at 100 SNPs that are each split 50-50 in the world's population? You might say, well, 100 SNPs of that aren't going to nail you down to a very small number of people. You're wrong. Because the first SNP is able to divide the world's population in half. The second SNP divides that half of world's population into quarters. The third SNP divides that quarter of the world's population into eighths. So you're getting down to 2 100ths of the world's population. 2 to the 100ths of the world population is a very small number of people. So yes, even 100 SNPs can tell you an individual's identity. All right. Some people have argued that publishing aggregate data protects us from this. If we instead say that these 10 people had this combination of SNPs, that you're breaking apart all the haplotype information, etc., and that's all good. This is worrisome and problematic, but it offers some amount of protection. Now, people have shown there are various attacks to this, what we sometimes call the hairbrush attack. I have a piece of DNA that I got from this person's hairbrush, and I'm trying to discern if they were in this study about this socially embarrassing disease. That's not the most common kind of identifiability challenge, but it's one that we need to be thinking about. So lots of nations have now published laws that deal with genetic privacy. Here we have the HIPAA legislation from the USA. We have the GDPR, I've already forgotten what that stands for, General Data Protection Restrictions? I'm not sure what that stands for. And the POPI Act here in South Africa. So if nations think it's important enough to create laws about doing this, you as a biotechnologist owe it to yourself to learn what is allowable for you to make available in your science publications in order to protect yourself and, more importantly, to protect the people you study. All right, this brings us to about the halfway point in the lecture, so I would suggest at this moment you hit the pause button and then we can all just take a quick run around. And welcome back. In this part, we're going to move to a rather different topic. We've talked now about how we would recognize that a variant exists in a person. We've talked about the value of those data. Let's talk, though, about how we discern if there's a phenotypic impact for protein coding genes. let's start as well with this idea that we have a taxonomy into which these variants fit. To simply say that the DNA is different at this site is one thing, but it's quite valuable if we could sort of put some terms on this. Often, you'll find that a gene variant might be called novel and non-coding. Novel meaning it's not in an existing database, and it's non-coding in that it's not associated with a protein coding gene. These are interesting, and yet, generally speaking, they're the hardest type of genetic variant for us to put any sort of phenotypic label on. So quite often, you're in for a round of many experiments in order to discern if there's a phenotypic effect for a genotype variant of this type. It might be that it is a known but non-coding variant. In a case like that, it might be that what you're looking at is an expression quantitative locus, quantitative trait locus, that this is a site known to be associated with gene regulation, meaning it might boost or diminish the amount of an accompanying gene product. So it might be that if you have a known variant that falls outside a gene, it might yet be important in how this gene is expressed. It might be that you're finding in-frame indels, which is to say something that falls within a gene that adds a codon or more of nucleotides, or that removes a codon or more of deletions. But if it's a case where you're adding 3 nucleotides, or 6 nucleotides, or 9 nucleotides, you're gaining amino acids, but you're not changing the frame in which that sequence gets interpreted. It might be that you have missense, a missense variant. And in a case like that, you've got some sort of non-synonymous amino acid change. A missense can do quite a lot to alter the way that this amino acid sequence gets read. You might, for example, create frame shifts. In a frame shift, maybe you insert a nucleotide or 2, but not 3, and that causes a reading, a difference in which codons are interpreted from the downstream nucleotides. So stuff like that can do quite a lot to make the C-terminus of a protein quite different than what it would be, even if you're adding a single nucleotide, if that's going to alter the reading frame for everything downstream and change what the C-terminus of the protein is. So frequently we have these two terms that show up then, an indel, which represents an insertion or a deletion, and we have this term non-synonymous, which is to say that we're not just changing nucleotide sequences, we're changing the amino acid sequence that results. Hopefully everyone remembers this diagram from perhaps high school. This is the genetic code. It took us a while to work it out, but it describes which combinations of 3 nucleotides lead to a change in amino acid sequence. So I would note that if we have A, U, G, we have a start codon. That's methionine as well. There's only one codon for methionine out of everything. But look at something like threonine. Here we have a set of 4 different codons that all result in threonine. If you were to change this last letter from U to C, or U to A, or U to G, it makes no difference whatsoever. Threonine is still what that codon relates for. What changes is essentially which tRNA brings the amino acid to the ribosome. So there are lots of changes we can make, particularly in the 3rd position of the codon, that would have no impact whatsoever on which sequence results from the process of translation. A non-synonymous SNV is an amino acid changing single nucleotide variant. This is a really important class of variant, and it's the sort of thing that just draws people's eyes when they're looking at a list of variants. Which ones are going to be easiest to interpret? The non-synonymous SNVs. So, what other changes did I want to say here? Yeah. So, these are sequence changes that alter which amino acid gets put in place. It's also the case that indels and missense variants are going to change the sequence, but they're not changing it in a very targeted way. They're changing like everything downstream. I would also note that it's possible to have a codon for tyrosine, say UAU, and replace that last letter with something other than a C, and now instead of putting in a tyrosine, we have a stop codon. So creating a premature stop codon has a rather dramatic change on the sequence, that everything after that is omitted from the translation. That's obviously a very big change. So generally, when we talk about non-synonymous SNVs, we're focused on these changes that result in one amino acid replacing another. I think one of the topics that students frequently lose track of in this is that we already know some reasons why substituting one amino acid with another could produce a relatively minor change in the functionality of a protein. Now, we've talked about Blossom 62, a substitution matrix, very, very useful for understanding over the process of evolution which substitutions of one amino acid for another are most common. But I would also ask that you think about it from a biochemical point of view. It might be that you replace a lysine with an arginine. Something like that is not likely to change the biochemistry of that site tremendously, because they're both basic amino acids. Arginine is far more basic than is lysine, so there's a change in magnitude basically, and there's a change in size, so you might have steric factors as well. But in general, if we were to see changes within these individual rows, changing a leucine to an isoleucine is they're not just the same mass, they're very similar in the kind of biochemistry they have on offer. So you might have basic amino acids, acidic amino acids. If you have a glutamic acid, a glue, replaced by an aspartic acid, they both have carboxylic acid side chains. Therefore, you're not going to get a huge swing in the biochemistry between the two. If you replace an asparagine with a glutamine or vice versa, they both have amides as side chains. So we see that there's quite a few things. I want to note down here that this last pairing is not the way you would normally see it in a biochemistry textbook. But from the point of view of how proteins function, we find that both glycines and prolines are associated with loop regions of proteins. So replacing one with the other is less likely to have a big impact than if you replace, for example, a proline with arginine. Quite different in their biochemistry there. So you can think about it from the point of view of a substitution matrix, or you can think about it from the point of view of biochemistry. Very frequently, you will find that people rely on software to discern whether a particular non-synonymous SNV is deleterious or pathogenic or disruptive of function, whatever. There are lots of different terms that get thrown at this. One of the earliest of these was SIFT, which is very valuable because it starts with the sequence the sequence context in which this change takes place. It's able to pull down sequences that are similar to that from a sequence database, align them, and compute the probability that this substitution would be tolerable or not. So this is essentially the same approach as what you'd see from the Blossom 62 approach. Polyphen was produced just a year later. It added some 3D structure elements to this and a feature table to say which parts of this sequence are, for example, the active sites. So the Polyphen software is quite valuable at bringing in functional annotation on the protein sequence, not just the sequence itself. SNAP, published in 2007, added some really cool stuff on solvent accessibility. This is a term that frequently comes into play when we talk about a protein structure, to say, is this sequence folding into a structure that makes the polar stuff on the outside and the hydrophobic stuff on the inside, and so on. So it gave us quite a lot of power to increase the amount of information brought to bear in deciding whether a non-synonymous SNV was going to be disruptive of function or not. These three algorithms have been joined by a pile of others, and in recent years we've seen a very large use of machine learning based methods to try to bring the best combination of scoring factors to bear in deciding whether this non-synonymous SNV is key or not. I would point out that over time we have found that protein structures which is to say solved structures of known conformations that sequences take on in solution, or in membranes for that matter, are becoming hugely valuable for us as a resource, as we've got tens of thousands of these things, hundreds of thousands of structures to work from now. It's becoming more possible for us to discern whether changing a given amino acid is acceptable or not. Threading represents kind of a personalized medicine way to think about these. If you know that the wild type sequence folds like this, and you change an amino acid right here, how would that alter the structure that results from it? So this is an attempt to merge experimentally determined structures with computational alterations to those structures that represent how that amino acid change would affect the conformation. So there are a series of steps that we go through. Most of these threading algorithms are kind of generational in aspect, which is to say that they build a model reflecting the altered side chains and then try an energy minimization on it to let it sort of settle into a preferred conformation, and repeat that to see if any further changes take place. So this iterative process hopefully gives us a sense of how this structure relaxes into form once we've made an alteration to the side chains within it. From here, I want to take a rather abrupt turn into the fine world of GWAS, of genome-wide association studies. This is a rather older approach now, and yet we still find that studies get published in this space. At the time that I was leaving grad school, these were catching fire in a huge way. So let's talk a little bit about this concept of linkage and genome-wide association studies. In the previous section, we were talking about the direct impact that if we know that a non-synonymous SNV takes place in this protein-coding gene region, and it alters this amino acid to that one, we were looking for direct effects. This variant changes the protein sequence, that changes the structure, that changes the function. That's quite different than what we get out of a GWAS. In a GWAS, we are likely to find a particular marker is associated with disease risk, for example, and from that we might learn about the genetic regions that are taking a role in disease. So in this case, we may find that a marker is associated with Parkinson's disease or whatever, and then we have to ask ourselves the question, what comes next? Do we say, because there's a particular gene 1 megabase away, that this marker is associated with this, that this gene, in effect, is playing a role in disease? A lot of papers made that kind of mistake. And so we need to be really careful that we're remembering that we're finding a region of DNA that's likely to be associated with our cohort definition. So the genome-wide association studies usually start with hundreds of patients. In this case, if we've got 100 patients with disease and 100 patients without disease, can we determine a set of genomic markers that help us to infer a new person's disease risk? So I would note that Klein et al. Back in 2005 were looking for age-related macular degeneration markers. They had 96 cases and 50 controls. In this case, they were using a relatively old kind of microarray that could measure 116,000 SNPs. So I would note they're not using sequencing. They're not using exome sequencing. They're not doing whole genome shotgun. They're just monitoring the genome at these 116,000 locations. They're not getting any information about places outside those 116,000 locations. They were able to then evaluate which of these SNPs were statistically significant in separating the 96 cases and 50 controls, and they found 2 that were to be associated. This opened the floodgates. This paper in Science led to 100 imitators with 1,000 different diseases. So when we look at how they did their study, there are a lot of statistical concerns that come into play. as I noted, in this case, they were looking at 116,000 locations in the genome and trying to discern which of those were associated with disease. One of the key things that really fell out of this paper, that this paper popularized in this way, is called the Manhattan plot. The idea being that Manhattan is a city full of skyscrapers and that some of them stand out way above the others. So in this case, we're seeing that each of these lines, each of these vertical lines in this plot, represents a different genetic marker. The ones that are very tall represent those with very low p-values. Now, I think you might remember that when we talked about Phred scores for base calling, that a very low probability of error yielded a very high Phred score, because the Phred scores are expressed on a negative log scale. They use the same trick right here. The probabilities are shown as the negative log base 10 of a probability, or of the p-value in this case. So these very high lines that we see at this location and this location represent very low p-values, that these two genetic markers were attributable very strongly to the difference in the two cohorts. All right, now what is all this business with multiple testing here? When we evaluate 100,000 different genetic markers, we're performing 100,000 statistical tests. That is not a great situation, because your chance on doing a t-test for two distributions where the numbers come from the same distribution is 1 in 20 that you're going to get a significant p-value, 1 below 0.05. The authors knew that, so they needed to have some way to adjust for that. They've made use of something called Bonferroni, the Bonferroni correction, as a way to deal with that. Here you see that they use data from 103,000 of their genetic markers, and they said, we're going to divide 0.05, which was our threshold, the 1 in 20 p-value threshold. We're going to divide 0.05 by 103,000. Then we're going to say only p-values below that very small number are going to be those we claim as significant hits. That's a really strong kind of multiple testing correction. In effect, the Bonferroni correction is a way of saying that we want to protect ourselves from making any false claims. That's different than another technique we're going to talk about with gene expression, the FDR technique. So here we're trying to protect ourselves from claiming any genes are significant when they're not. So this approach meant that they could put this horizontal line across this graph. You see that showing up just above 6 on the vertical scale. That is to say that they computed a p-value threshold of 4.8 times 10 to the negative 7th was how low the p-value had to be before they could consider it significant under these circumstances. And as you see, only those 2 points fell above it. So this approach of Bonferroni correction and Manhattan plotting shows up all the time when we look at GWAS data. Let's talk a little bit more about markers and linkage. We talked before about how haplotype is something that we care about, which is to say that each genetic variant isn't independently drawn from a given person. If you look at rare genetic variants, we find that they are typically in the company of other variants that are nearby. They tend to co-segregate through the population together. The reason for this is that recombination doesn't have a lot of opportunity to separate them. You remember in the process of making gametes, our DNA crosses over to form these recombination events, thus exchanging a bit of the paternal DNA with a bit of the maternal DNA on a chromosome. Right, so in a case like this, we can note that particular SNPs, when found to be sequentially arranged across a piece of chromosomal DNA, are frequently found to be in linkage. In this case, we see that 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 different SNPs that are all attributed to this region of this chromosome hardly ever get separated from each other. This area of really, really high linkage is just it's not it's the opposite of a recombinational hotspot. On the other hand, we find that these markers down here tend to stay together. And they stay together in a way that this section down here and this section over here don't get broken up, and yet the area between them is a much more likely place for recombination to take place. So linkage is important. It might seem that two markers are quite close together on the DNA, but if recombination between them happens all the time, they're not likely to be linked when we look at them in population studies. So this topic of linkage also matters when we're trying to discern an identity to what this linkage means. If I knew, for example, that this marker were very strongly associated with my phenotype of interest, and this gene falls quite close to its linkage region, I might feel more secure in saying this linkage, this marker is indicative that this gene is involved in that process. But I would really need to know the linkage structure of that area before I could make a claim like that. Lovely tea. So it's always worthwhile to remember that these operations that we're doing on genetic data require quite a lot of software to make them happen. And Planck is one of the most widely used software created for this purpose. In this case, it's implemented in a language called C++, which often produces rather high-speed executables. So if you have a very computationally intensive task, you might implement it in this. It's one of the big packages that we use for GWAS data analysis. Here I was looking at the particular features that were integrated into one of the more recent releases of the software. You can see that they focused on data management, high-speed summary statistics that were parallel computed, population stratification, association analysis, and identity by descent estimation. These are all the kinds of operations you would really need to do to understand a GWAS data set. Imagine that you're doing a study that involves 400 people and you fail to take into account that these 3 people are all part of the same family. That would be really awkward and embarrassing. So the software is going to handle things like that for you. The stratification issue is a really huge one. We're going to spend a moment talking about that in some detail in just a moment. But I would note that these tools have been engineered to run under Linux, Windows, or Mac OS X. There are even some GUIs that have been created, a graphical user interface to make it easier to talk to the software. So you don't have to be a command line jockey to make use of tools like this. Okay. I've mentioned that genome-wide association studies are enormously popular, at least they were enormously popular. As a result, we see that folks like the European Bioinformatics Institute have gone a long way towards creating catalogs to help us to understand what we know about a given piece of DNA. Here we're looking at something like a karyotype. Essentially, we're looking at each chromosome drawn out separately. So looking at a particular chromosome, we might ask what associations have been made with the DNA falling within this 10 megabase stretch. And from that, you'd probably come back with multiple GWAS studies that have all implicated markers within that region to be attributable to a particular disease. So this helps us to look across papers, in effect. If you think of each of these colored balls as a paper that's studying the GWAS of, that's made a declaration about this particular region of DNA, you're able to see that some parts of the DNA are very heavily covered and others much less so. Okay, but in any case, if you want to know what genetics associations have been made for a given disease, the GWAS catalog is a really good place to just go look that up. I mentioned that stratification is a really big problem that frequently goes uncorrected. Well, it used to be frequently uncorrected in GWAS studies. These days, if you try to publish a GWAS study and fail to take stratification into account, you're quite likely to get your ears knocked about a little bit by the reviewers. And they'd be right to do so, because stratification is a very widely understood problem at this point. Let us imagine that we're trying to find the difference between a pair of cohorts, but our cohorts are not homogeneous in population. You can create problems for yourself in that the differences in the populations of people that you've used for the two cohorts can trick you into finding associations that are not real. So we speak of stratification as a systematic ancestry difference between cases and controls. So here I've drawn the example that I have a case control study that I'm trying to do a GWAS on, but my cases were 60% Han Chinese, and my controls were 40% Han Chinese. If I just push forward with Plink and don't tell it anything about the population, allow it to assume that stratification is an issue, I'm going to come up with a bunch of fake associations. The reason for this is that an uninformed analysis would assume that Han markers are indicative of disease. You see that? I say, well, 60% of my Han Chinese, my cases were 60% Han Chinese, my controls were 40% Han Chinese. In effect, this is saying that the genes, the genetic markers associated with being Han Chinese are 50% higher in the cases than they are in the controls. The software could make the mistake that, having the markers of being in the Chinese population is associated with disease. Big problem, very big problem. So one of the ways that we deal with this is to try to infer from the genetic markers what ancestry each of the people in our study can have within them. And this is not something where we say, this person is from an African population, and this one's from an Asian population, and this one's from... nonsensical. In the kinds of stratification we're looking for, each person is going to have multiple contributors that lead to the genetics that they have. So being able to recognize the multiple lines of ancestry that feed into each person becomes quite necessary if you're going to pull any sort of useful information out of this study at all. So this raises the topic of admixture. I think everyone understands the history of the world is a little complex, and over the course of the last few hundred years, we've seen people from all sorts of different locations on Earth come together in new locations and make families. This creates a situation that we describe as admixture. So populations that came about by the mixing of two or more distant continental populations in recent history exhibit both fine scale and segmental scale variation. So one obvious example of this in the United States, where I'm from, is that a bunch of people were brought against their will from the coast of Africa to work in plantations throughout the South. They, throughout a lot of the Western Hemisphere for that matter. They went on to have kids, and they went on to have kids, and they went on to have kids. Today, when you draw a random person out of the population, you're quite likely to find that there's some mixture in what ancestry they have. Admixture is the way that we evaluate the contribution of each of these different populations in each person. Okay. So a really interesting study on this was performed back in 2010, and it helps to illustrate just how difficult a problem it is to go studying genetics in South African populations, because South Africa is the example par excellence of people coming from all over the world to create the country that we have today. So in this case, they were trying to evaluate the genetic background markers that can complicate doing studies in the South African colored population. We know that there's a wide variety of groups that have come together to create today's colored community within South Africa. So in this case, we're looking at the same people sorted 4 different ways. So each stripe through each of these panes represents a different person. We're now sorting this person by sorting the people in this first case by the number of markers associated with being part of the Khoikhoi or San populations. So this person at the left is the person with the least markers associated with Khoisan. This person at the right is the person who had the most associated with Khoisan. But sort the same data a different way, you can ask about the degree of the black African markers that were found in individuals. We see that one example over here at the right had a really high level, something like 90% of the markers were associated with being from the black population. Some of the folks within the colored population have 0 on the other hand. We can ask about European markers within these populations, and we can ask about Asian markers. We had lots of people who came here from areas in the Far East, in the Spice Islands and so on, that ended up here in South Africa. So we see that all of these different populations have come together to create this population today. If we fail to take that into account and try to do a study for, let's say, diabetes risk in the South African colored population, we could make a really big mistake in associating the disease with one of these 4 incoming populations rather than understanding the markers that this individual has that are not associated with his or her racial background, but rather the markers that are not associated with it. So there were a lot of markers, a lot of different points that we wanted to raise in today's talk. I'm going to try to just boil all of that down, to distill it down to just a few key messages here though. Genetic variation can be measured through whole genome shotgun, we know that. It's expensive, lots of data management problems as well. It may be studied by whole exome sequencing, which helps focus the attention on just the 1% of the genome that's known to be associated with protein expression. Or you can use microarray or bead array technologies to get an assessment of genotype on the basis of well-known markers that are part of these sets. All of them have their strengths, all of them have their weaknesses. It's not a clear-cut case that, well, if you can afford it, you should always do whole genome shotgun. That is not the case. And at many points, you're going to find that your information is much more targeted if you choose an option that lies between those. Non-synonymous SNVs draw particular attention among all the different variants that we can find because they have a recognizable effect on protein sequences. There's quite a lot to be said on how one discerns whether a non-synonymous SNV affects phenotype or not. I hope I've helped to guide you through some of the basic tools that we use for this purpose. It's probably a mistake for you to simply use SIFT because that's an old one and everyone knows how to make sense of it. There are lots of these phenotype detection engines to work with, and you may find that they disagree on which of these markers are actually associated with a big change in function. Linkage correlates a variant to phenotype, but it does not claim that a particular variant causes that phenotype. This is one of the things that most frequently gets my ire if I'm watching a research talk at the end of the year. That people who know a particular genetic marker is correlated with a functional difference, but somebody who hasn't pinned down any mode of action by which this variant can cause that phenotypic change. So lots of messages, but detecting genetic variants and understanding their effect on phenotype is a really popular place to be working, and I hope that this has helped you become a little bit more familiar with the tools that let you achieve that. Thank you very much.